Imagine a loved one is suffering from colorectal cancer and has to go in for a relatively standard procedure to resect part of their GI tract. A week after the operation, their condition is getting worse. The physician who's concerned orders a CAT scan, which comes back bearing terrible news. The connection formed during the procedure is now leaking. Bacteria from inside the GI tract is getting into the peritoneal cavity. Unfortunately, this is the case of up to 30% of resections. If untreated, it will result in multi-organ failure and possibly death. Even if treated, it will require a significant increase in the length of stay and tens of thousands of dollars in additional care. So let's go back to the procedure to understand exactly what happened and how something like this can be prevented in the future. The goal of a resection is to take out a diseased portion of the GI tract. It starts with the physician stapling and cutting in order to separate out this area, which is then able to be removed. After, the remaining ends are connected, forming an anastomosis. So what caused the leak to occur here? We spoke to physicians across a range of disciplines, and after weeks of observations and clinical feedback, it was apparent that there are three main reasons. Poor perfusion at the resection site, a mechanical leak or gap in the sutures, and tension at the connection. Evaluating both the importance of these factors as well as the availability of current technology, we recognize that perfusion at the resection site was essential and that there are many shortcomings with current technology. Current technology involves injecting an ICG drug intravenously, which fluoresces when looking through a special camera. This requires large pieces of capital equipment, which are expensive and not well integrated into the workflow. And for that reason, they're not often used.
In order to address these shortcomings, we've developed GuideOx. GuideOx is a smart surgical stapler that allows physicians to intraoperatively test for healthy blood perfusion at the resection site. Current staplers work by first clamping down on the tissue in question and then firing staples and cutting, sealing both lines as it cuts. Whereas our stapler will measure perfusion once it's clamped down and provide that feedback to the physician before stapling and cutting. We're able to do this by leveraging current technology used inside pulse oximeters. Pulse oximeters use a technique called PPG, which measures the absorption of light as it passes through the tissue, allowing us to measure blood perfusion within the region. How this will work in practice is the surgeon will place the surgical stapler like normal. GuideOx will then provide quantitative measurements alongside a red or green indication. A red indication will mean there's poor perfusion at the site, and the surgeon will find a new location. A green indication will mean that there is good perfusion, and this is an optimal location, and the surgeon will go ahead and make the cut. The surgeon can then repeat this process on the other side, completing the resection with more confidence that an anastomotic leak will not occur. GuideOx will be a disposable, one-time use surgical stapler, like the current surgical staplers on the market. This will allow it to easily integrate into the current physician workflow, unlike our competitors, which are expensive capital pieces of equipment that require the additional use of fluorescent dyes. Most importantly though, GuideOx will provide a quantitative measurement, allowing physicians more assurance when selecting an optimal resection site.
GuideOx provides value to the patients by decreasing the morbidity and mortality associated with anastomotic leaks, to physicians by being directly integrated into the workflow and reducing the likelihood of reoperation, and to providers who often have to cover the cost of additional care associated with an anastomotic leak. Leaks most commonly occur in the lower GI tract. There are over 320,000 colorectal resections done in the US alone each year. Looking at both the price of current staplers and the addition of our technology, the resulting market is $640 million. Although our initial focus is on lower GI, addressing procedures throughout the GI tract will expand our market to over $1.1 billion. In terms of FDA approval, we will be pursuing a Class 2 510K pathway using other linear staplers as our predicate device.
So, as far as big picture for key milestones in funding, we hope to secure $500,000 in funding after filing a patent this year, and that will cover prototyping and IP development. After that, in year 2, we're hoping to secure $5 million to cover P&V, clinical After that, in year 3, we expect our first year of sales at Texas Medical Center, which we've already been building relationships with. After our product's proven success there, we expect rapid growth, which is why we're asking for $10 million in year 4 to cover the sales that we expect. By year 6, we expect a cash flow positive status. In order to bring this device to market, we put together a diverse team with backgrounds ranging in electrical, mechanical, biomedical, and business backgrounds. We're also supported by advisors in medical entrepreneurship and device innovation at Rice, Baylor, and Johnson & Johnson. So what we're asking from you is threefold. First, clinical advisors in colorectal surgery. Second, funding to help support research and product development. And third, technical advisors who have brought medical devices to market. We are Guidehouse. Let us guide you to the best results.
Okay, 4 minutes of Q&A.
Do you have any type of prototype at this point?
Yeah, so currently we have some initial benchtop testing data. Also, we're working with Johnson & Johnson Center for Device Innovation and using their expertise to continue prototype development, as well as running some animal studies. In 3 months, we hope to have the results from those animals.
What makes you believe you've got a patent opportunity?
So we're working towards a licensing agreement with Brightseed Medical, who's actually a previous RBPC company, and they've expressed a lot of interest. Jonathan Dunn has expressed a lot of interest in terms of working with us and Johnson & Johnson Center for Device Innovation in order to continue product development. Because this is a unique application of the technology, there's also a possibility to generate, or there's a probability that we'll be able to generate our own IP.
When do you think your earliest exit opportunities are, and to whom?
So that's a great question. There's a lot of factors that definitely go into that. I'd say that one of our huge current advantage right now is we already have a working relationship with Ethicon. They've expressed interest in this type of technology and integrating it into their staplers. In terms of the earliest ability for us to exit, they would definitely want to see positive clinical or animal evidence at least. And that's really dependent in terms of how they see our value and whether or not competitors also see value in that regard.
So you mentioned Ethicon. Are you involved with CDI now? Do you have a development agreement with them?
Yes. So CDI has been working with us closely in terms of the initial clinical needs finding process and then developing a solution. They've been advising us in terms of what would be well integrated into the Johnson & Johnson pipeline. So Ethicon and Medtronic are the two major players when it comes to linear staplers. And they see this product fitting very well into Ethicon's portfolio. At the same time, Ethicon is, as I said earlier, expressed a lot of interest.
But is your CDI relationship formalized yet or informal?
It's a formalized.
It is formalized?
Yeah.
Okay, thank you.
Great presentation. Curious, if you can't secure a license from Brightseed, what happens?
Sorry, you said if we cannot.
If you cannot secure a license.
Cannot secure a license from Brightseed.
What happens? Because it seems like the technology really kind of bumps up into their technology stack.
Yeah, so Eddie can talk a little bit more about the other options in terms of different ways to measure perfusion at the end of the stapler, which is Brightseed's main patent is around the fact of using a light and a sensor, and then there's a lot of other ways in which that can be done, in which that would be our patent.
So other ways of measuring perfusion could be through thermal transfusion, hyperspectral imaging, and ultrasound. Specifically for our application, ultrasound isn't really feasible due to the limited area in the abdominal cavity. So we would either pivot towards more of the hyperspectral imaging route, as well as looking at thermal transfusion.
So if you have to go that route, then what kind of domain expertise do you have to drive to do so?
In terms of hyperspectral imaging, it would be more focused on data algorithms, capturing each digital signature from the spectral wavelength, and then having an algorithm that to compile all the tissue structures that we see in our image and identify where perfusion is.
That's